Hey guys, in today's video we're going to be discussing the structures of Cas9 and the steps that occur as Cas9 edits the genome. So before we get further into the video, I want to briefly discuss with you what CRISPR Cas9 actually is. So CRISPR Cas9 is a gene editing tool that can be programmed to target and locate specific genes in a DNA sequence and just cut them out. So an example of this would be me. I have curly hair. So if my parents didn't want me to have curly hair when I was born, basically using CRISPR Cas9, they could go in there, locate the curly hair gene and cut that out and replace it with whatever gene that they want using guide RNA. Okay, so starting off, we're going to talk about the structure of the CRISPR Cas9 protein. So first you have your Cas9 recognition lobe. That's going to be what mediates the DNA strands and kind of finds the site where editing is going to occur. Down here you have your Cas9 nuclease lobe. Those are going to act. That's going to act as the scissors of the protein and is going to cut out the spot that's going to be edited. So now that we've discussed the different components of the Cas9 protein, we're going to be getting into the three main steps here that we're going to be talking about. The first is going to be genome editing. The second step is going to be the knockout. And thirdly, we'll go over transcription control. Okay, so here we have our guide RNA strand here within the Cas9 channel here on our recognition lobe. So our recognition lobe has found the area in which our target DNA is, our target strain is going to be. So typically what's going to happen here is that Cas9, the protein, is going to enter a nucleus and it's going to lock onto a short sequence known as PAM. So here's PAM down here, and in the next clip I'm going to show you that when the DNA is going to bind to it. So here's the PAM recognition domain, and here's the PAM sequence that Cas9 has found. Alright, so now what's going to happen is this DNA is going to line up with the guide RNA strand, and once this is done, the DNA is going to unzip, and then this Cas9 nucleus lobe is going to come in and it's going to make a double banded break in the DNA strand. So now we have a double-stranded break. Now, a double-stranded break occurs when both the strands are now cut. This is formally known as non-homologous end joining, or NHEJ for short. So non-homologous end joining occurs when there is a double-stranded break in the DNA sequence, and there's an alarm kind of being set off because DNA wants to repair that break that's been made. The DNA doesn't really know necessarily if there's been anything added or taken away from the sequence, so this often causes a mutation within the DNA sequence. So here's our repair template, or the DNA's repair template rather, that is going to fix this break that we have made in the DNA using Cas9. And just like that, bam, now we have our new DNA sequence with our new base pairs located here where the break is. Now for the knockout. So here we have our guide RNA placed alongside the Cas9 recognition lobe within the Cas9 protein channel. And again, so now you have your DNA sequence lined up with the guide RNA. You have your PAM sequence found by the PAM recognition domain on the nuclease lobe. And so now you're ready to cut. And just like that, a cut was made by the molecular pincers on the nucleus lobe. This causing a double-stranded break in the DNA sequence. Now, similar to last time, this is our knockout. Or I'm sorry, this is going to be our insert, which is going to be placed in where the break was made in our DNA sequence above. So here you have your finished product. You have your base pairs inserted within the break that was made inside the DNA sequence. So here's your new strand. Here's the start of our transcription activity. So here we have our guide RNA placed within our Cas9 channel here on the recognition lobe, which is going to find out where the gene editing is going to occur. So here we have our DNA within inside the Cas9 channel lined up with our guide RNA strand here. We have our PAM lined up with our PAM recognition domain, little PAM sequence, and for purposes, because I don't have tape, we're just going to pretend that our transcriptional activator or repressor is actually up right now, allowing the DNA to come into the channel. Okay, so now that the DNA is in there, our transcriptional activator or repressor is going to bind to the target DNA, which is either going to turn the DNA on or off, depending on the situation. Alright guys, so that was our last step. We have successfully edited a gene. We went through the phases genome editing, the knockout, and transcription control, as you just saw in that last step there. Thank you guys so much for watching. Hope you guys enjoyed the video and you learned something. Stay curious. I'll see you guys later.